So we're going to talk about diuretics in this talk. Okay, so this is a very important topic in pharmacology. So these are a list of possible learning objectives. So this list is taken from Kazoo. It's quite a reasonable list for you to target your studying and be more focused when you study, because you are studying to answer these questions. Okay, that is so this list will help test you. Okay, whenever you read something, it will help you test yourself and see at what level are you in your mastery of the topic. So basically we have drugs used in kidney disorders. Okay, so this is the big topic. And then this is again from Kazoo. So we have drugs used in kidney disorders, and then divided into two: drugs that alter or modify salt excretion, and then another group is drugs that alter or change water excretion. Okay, so one is salt, okay, sodium chloride, and then the other one is water, okay, water excretion. Salt, water. Okay, so and then you can see that this categorization is quite to a certain extent anatomical. So from the proximal convoluted tubule to the ascending loop of Henle, the distal convoluted tubule, and then to the collecting tubule. Okay, so at the proximal convoluted tubule we have mainly the carbonic anhydrase inhibitor. Okay, the carbonic anhydrase inhibitors, mainly acetazolamide. So this drug is not so important for this particular topic, diuretics, but it's mainly used for other purposes. And then we have the loop diuretics, okay, acting at the ascending loop of Henle, mainly furosemide and torsemide. And then we have drugs that act at the distal convoluted tubule, the thiazides, mainly the hydrochlorothiazide, chlorothiazide, and chlorothalidone. And then we have the potassium sparing diuretics, okay, potassium sparing diuretics, spironolactone and amiloride, so very important drugs, okay, potassium sparing diuretics at the CCT. And then we have drugs, this category that acts both, modifies both salt and water excretion. So these are the osmotic diuretics, okay, osmotic diuretics. Example is mannitol, okay, mannitol. And then the others are ADH, antidiuretic hormone agonist, such as desmopressin. And then we have the ADH antagonists, such as conivaptan, okay, conivaptan. Okay, the diuretics. So diuretics are drugs that increase the volume of urine excreted. Okay, it increases the amount of urine that is excreted from the body. And most of these diuretics are blockers. They are antagonists. Okay, they hinder or they block renal ion transporters. Okay, they block renal ion transporters. So what they do is they basically decrease the reabsorption of sodium at various sites in the nephron. Okay, so we have many sites in the nephron, at least 5 main sites in the nephron. So these diuretics will reduce the reabsorption of sodium at these various sites, and this will lead to sodium and other ions such as chloride. Okay, sodium and other ions such as chloride to enter the urine in higher than normal amounts together with water. And this will all be carried passively to maintain osmotic equilibrium. So diuretics hence increases urine volume, okay, and often it changes the pH and also the ionic composition of urine and blood. Okay, that's how powerful diuretics are. So they can, besides increasing urine volume, they also can change the pH and the ionic composition of both urine and blood. Okay, remember that. It can alter the pH and the ions that are present in the urine and blood. So we have various classes of diuretics, and the effects of these different classes, they can differ significantly. They can they can have various, have a wide range of effects. Okay, so we can have from the increase in sodium secretion, which is less than 2% for the weak ones. Okay, the weak diuretics, the potassium sparing diuretics. So the potassium sparing diuretics, they are weak in terms of how much sodium can be secreted out, okay, can be thrown out. And to the stronger diuretics, the more potent loop diuretics, which can excrete more than 20%, okay, of sodium. So loop diuretics. So from the ranking, so in terms of diuresis power, loop diuretics are the top, okay. So the potassium sparing diuretics are quite towards the bottom. Okay, so the main use of diuretics is mainly for treatment of edema. Okay, and then we have management of edema and treatment of hypertension. So before going into diuretics, let's talk about the normal regulation, the normal control or maintenance of fluid and electrolytes by the kidneys. Okay, so about 18% of the blood plasma, about 20% of the blood plasma entering the kidneys will be filtered. Okay, will be filtered from the glomerular capillaries into the Bowman's capsule. And the filtrate, even though free of proteins and blood cells usually, they contain most of the lower molecular weight plasma components in concentrations similar to that in plasma. So even though it does not contain proteins and the blood cells, okay, but because it contains most of the lower molecular weight plasma components, so it looks a bit similar to the plasma. Maybe the concentrations are a bit similar to the plasma. And these include glucose, sodium bicarbonate, amino acids, and other organic solutes, okay, and also the electrolytes, sodium, potassium, and also chloride. Okay, so the kidneys control the ionic composition, okay, the ionic composition and also the volume of urine by active reabsorption or secretion. Okay, so it's either active reabsorption or secretion. Okay, very two basic mechanisms. So it's either active reabsorption or secretion of ions and or passive reabsorption. Okay, passive reabsorption of water at the 5 functional zones, okay, along the nephron. Okay, now we're going to go into the 5 functional zones. Okay, number 1 is the proximal convoluted tubule. Here almost all the glucose, bicarbonate, amino acids and the other metabolites are reabsorbed. And about 2/3 of the sodium is also reabsorbed in the proximal convoluted tubule. And then we have chloride, okay, chloride passing into the lumen of the tubule in exchange for an anion such as oxalate, okay, as well as paracellularly through the lumen, okay. And water will follow passively from the lumen to the blood to maintain osmolar equality, okay. So we know that sodium is reabsorbed, but chloride will pass into the lumen, okay. And then we have water moving from the lumen to the blood, okay. It's being reabsorbed, okay. It's because it just follows passively according to maintain the osmolar equality. And the sodium that is reabsorbed, okay, so we remember that sodium is being reabsorbed. So here we remember 2/3 of the sodium is also reabsorbed. So when sodium moves, of course, water will usually tend to move together. Okay, so the sodium that is reabsorbed is pumped into the interstitium by the sodium potassium adenosine triphosphate pump. Okay, and this will lead to normal levels of sodium and potassium in the cell. So we have carbonic anhydrase in the luminal membrane. Okay, carbonic anhydrase in the luminal membrane and also the cytoplasm. Okay, besides the luminal membrane, also in the cytoplasm of the proximal tubular cells. So this will control the reabsorption of bicarbonate. Okay, carbonic anhydrase is responsible for and for controlling the reabsorption of bicarbonate. So next we talk about the organic acid. So next we talk about the organic acid and base secretory systems. So the proximal convoluted tubule is the site of the organic acid and base secretory systems. Okay, so we have the organic acid secretory system, and then we have the organic base secretory system. Okay, so the organic acid secretory system, it will secrete, okay, a variety of organic acids, example uric acid. Also it secretes some antibiotics and also secretes diuretics, okay, from the bloodstream into the lumen. Okay, this lumen will lead to urine. Okay, so we have urine that is going to finally be excreted out from the urethra. Okay, so and most diuretic drugs will be delivered to the tubular fluid via this system. Okay, and this system is a saturable system, and diuretic drugs in the bloodstream usually will compete. Okay, it will compete for transfer with endogenous organic acids such as uric acid. Okay, in the next slide we will show you how probenecid affects penicillin secretion. Okay, so that's organic acid secretory system, and then we have the organic base secretory system, which controls secretion of creatinine and choline. So this is basically the diagram showing how penicillin. Okay, so we have blood here, and then we have the renal proximal tubule cell, and then we have urine. Okay, so we have the basolateral area and the apical area. So penicillin and uric acid, so they will compete, and then they both can be secreted here. Okay, they can be secreted here. They can be transported via the OAT1. Okay, so the anionic transporter system. Okay, so organic acid transporter system. So it can move this inside here and goes to the urine. And we have probenecid and aspirin, among others, that can block this. Okay, it can block this. So when you have probenecid, or if you have aspirin blocking here, okay, when you're both taking penicillin and you're taking probenecid at the same time, so potentially probenecid and drugs like aspirin can block this, so that you have penicillin being in your body for a longer time. Okay, so its action will be prolonged. Similarly, uric acid you have problems getting rid of uric acid, so uric acid will not go here and go into the urine. It will not be excreted. Okay, so that's a potential problem there if you take these drugs for a long time. Okay, next place is the descending loop of Henle. So the remaining filtrate, okay, after going through the PCT, it passes through the descending limb of the loop of Henle and enters the medulla part of the kidney. Okay, we have the cortex and then the medulla, and the osmolarity increase along the descending portion. Okay, the osmolarity increase along the descending portion of the loop of Henle because of the countercurrent mechanism responsible for water reabsorption. So you can ignore this term. You just remember there is water reabsorption happening along the way, so that becomes more concentrated, okay, downwards. So this causes the tubular fluid with a 3 times increase in salt concentration. Okay, do not need to bother too much about this. Just remember it's the concentration of salt becomes higher because there is more absorption of water happening along the way. So osmotic diuretics manifest part of the action in this region. Okay. Okay, next we'll continue and talk about the ascending loop of Henle. Okay, ascending loop of Henle. See you in the next video.